Okay. So I want to give you a little overview of fungal diseases. So this is what we study in my lab, and the reason behind it is their significance across the world. And so there's a large impact of fungal diseases, whether it be like in systematic infections or more invasive infections that impact over 1.5 million people. And what's most notably is the mortality rates are really high. So between 15 to 65%, they actually go up to 90% depending on the area and the availability of different drugs. And so one of the reasons behind this is because we have a difficulty in surveying for fungal diseases and fungal pathogens, as well as diagnosing them and teasing them apart from commensal fungi versus pathogenic fungi. There's also an emerging increasing rate of fungal disease across the world with higher rates of climate change and warming climates where they can adapt. And if you've seen The Last of Us and you see that progression that occurs, in real life, it would actually be far worse than that. And so I watched that show and I'm like, oh, I can't even think about it. So, but it's something, it's important for us to study and learn about. And then the WHO has listed critical priority groups of fungal pathogens. Cryptococcus neoformans, the one I'll speak to you about today, is one of the highest ranked ones. And that's because of its changing environmental and geographical locations, as well as host differences, and the rising rates of antifungal resistance. And so some of these drivers is what I'll talk to you about today and how we can overcome it and what we're doing in the lab and how we're using mass spectrometry to really improve our ability to fight these pathogens. And so from Marie's introduction, which was lovely, I really do work to try to have that interdisciplinary view of our research program and how things work together and pull from different areas. And so often I categorize our work in our research program as a One Health perspective to tackle different global problems. And One Health is the integration or the connections across animal health, human health, and the environment. And so how does a pathogen adapt to a host? How does a host defend itself from a pathogen? But then how can we also get novel antimicrobials, for example, from environmental sources? How can we design new therapeutics? And how can we use plants or environmental resources to produce these therapeutics? And so this is where it all comes together in that One Health context. And I find too providing an example of how we study One Health to be rather useful. And so this is one of the examples that we use in my lab. Half my group studies fungal disease in agriculture and plant environments, and then half studies more in a clinical or human environment. And so for different diseases, such as Fusarium graminearum, this is a fungal disease that affects cereal crops. Prevention of the disease is caused by annual fungicide applications. And azoles are the typical fungicides that are applied in the field to prevent these infections. The risk is that these fungicides, or the harm is that these fungicides are then released into the environment. And once they're within the environment, the microbes in that environment are exposed to them. And one of these microbes includes Cryptococcus neoformans, the fungal pathogen I mentioned before. And so Cryptococcus is now exposed to this fungicide. However, we also have inhalation of the fungus by humans if the environment is disturbed. And if you have infection and disease progressing, and if it's an antifungal resistant strain, that's where we have very limited clinical abilities to fight the disease. And so beginning from the field and the environment and crops and preventing fungal disease in wheat, for example, to then looking at the detoxification of mycotoxins and different toxins that are produced by fungi and how that impacts animal and human health, to moving more into how can we prevent infection of human fungal diseases, how can we detect infection once it occurs, how do we then treat infection, and if we have resistance, how do we overcome it? And so these are the different ways that we tackle this One Health problem in my lab using proteomics and bioinformatics. And so to highlight some of the work that we've contributed, specifically within the Cryptococcus realm, that is what I'll talk to you about today, really looking at more that health-driven aspect of what we're doing. To prevent, we have different antivirulent strategies. And here we're actually using snails and mollusks from the environment and looking at their extracts and then testing out the antifungal properties from these extracts. And it's amazing how different snails actually can protect from different fungi or different fungal properties. We then look at different diagnostic methods. And so we have constructed an organ atlas of cryptococcal infection to identify biomarkers from the host side and the fungal side and look at how disease progresses and can we target specific proteins along the way to detect infection. We've contributed a plethora of literature on the host pathogen interactions. And so this is looking at the fungi and macrophage, fungi within a mouse model system, as well as the co-infections between fungi and bacteria. And then finally, and what I'll focus on today, is this really reversing antifungal resistance. So if we have infection occurring and if we have resistant strains, how can we overcome it using proteomics and protein-derived approaches? And so Cryptococcus neoformans, it is present within environmental sources such as eucalyptus trees and pigeon guano, as well as decaying wood bark. And so my family and I were actually recently in Australia, and my kids wanted to hold a koala. And all I can think is like, oh, they're going to be infected with crypto, and like because they eat eucalyptus trees, and like it really ruins the moment for you as a mother and a scientist. And there's also geographical occurrence as well. So many cases of Cryptococcus occur within sub-Saharan Africa, and this is because of co-infection with AIDS, HIV and AIDS. This has decreased quite a bit over recent years with the introduction of antiretroviral therapies. However, we have now a spread of the infections. And so in the Pacific Northwest of the US and Vancouver Island, as well as in Australia, it is also endemic. And so that global distribution is increasing. If the fungal cells are inhaled from the environment, then a healthy or an immunocompetent individual can clear the pathogen from their lungs with the act of macrophages as well as T cells. However, for immunocompromised individuals that may not have those same immune responses or immune system capabilities, the fungi can lay dormant within the lungs, or it can also disseminate throughout the body, and it can cross the blood-brain barrier, leading to contaminating the central nervous system and causing cryptococcal meningitis. And so this is one of the most devastating outcomes of the disease and where we really need to improve our diagnostic and our treatment options. And so currently for treatment of cryptococcosis, we have 4 classes of antifungals, 3 that actually work for cryptococcus, and they have different modes of action. Azoles being the one that I'll speak to you about today, and the one that is used within the field, within the environment. And so that structured similarity that occurs and that's present is what is driving the resistance in the clinic as well. And for azoles being fluconazole, when we look at the treatment for cryptococcal infection, it starts with an induction phase. So we use amphotericin B and a cytosine, so a combination therapy. But this requires hospitalization, is highly neurotoxic to the patient. And so in many parts of the world, it is not possible to keep people in the hospital for 2 weeks undergoing this therapy, or the drugs are not available in areas. Then we have a consolidation phase where fluconazole is provided, and then a maintenance phase where patients are actually on fluconazole for 12 months or longer. And so you can also imagine that resistance from the exposure within the environment is a problem, but also exposure and resistance builds as the individual is treated with these drugs as well. And so we're taking an integrated approach to overcome antifungal resistance. So what we've done and what I'll talk to you about is looking at resistant strains and susceptible strains to this antifungal. We then use proteomics because I love proteomics and we use it for everything in my lab. We use proteomics to look at what are the protein differences between resistant strains and susceptible strains. And can we then target the proteins and the genes that encode those proteins and disrupt them to overcome resistance or to reverse it and make the fungi susceptible to the drug once again. And so to do that work, the first thing we did is we have evolved a resistant strain within the lab. And so the top graph here shows our wild type, which is the fluconazole susceptible, and our lab evolved, which is our fluconazole resistant strain. In the absence of fluconazole, they grow comparably. And in the presence of fluconazole, our resistant strain is resistant and can maintain growth. We then also looked at phenotypic characterization of this strain to look at the kind of the fitness effects that resistance can have on different strains. We see, so melanin is a pigment that is produced by the fungus to protect it from reactive oxygen species within the host, as well as UV light within the environment. And so we see that we have an increase in production of melanin within our resistant strain. If we look at growth and thermotolerance, they're fairly comparable between the strains. And then if we look at the capsule production, which is this polysaccharide halo that surrounds the cells, we also see that we have a significant reduction in the capsule between the resistant strains and the susceptible. So there's some trade-off for the fungi when it has this resistance that it's promoting. And so we take our strain, resistant and susceptible. We also added in a clinical isolate of Cryptococcus that is also resistant to fluconazole, and we performed proteomics on these samples. And so here in our principal component analysis, you can see we have our resistant lab-evolved strain here and our clinical isolate, and we have our wild type up here. So we have nice separation, whether it be our lab-evolved strain that came from the lab-associated examples or from our clinical isolate, which we expect because the clinical isolate will have confounding factors that drive protein level differences. And then we see we have separation from our susceptible and our resistant strains. We then do our multiple volcano plots, which as proteomics researchers, we love to do. And so here in the dot plots, these are representatives of such volcano plots where we show the significant increase of about 50 proteins in the resistant strain, a significant decrease of about 50 proteins in wild type. And then we have our clinical as well, where we see much more drastic differences between our clinical isolate and wild type. And then based on gene ontology, we also kind of look at the representation of proteins that are driving some of these differences to get an idea of what is going on in the strains. And so when we put all these together and we look at the proteins that overlap, our kind of our core proteome of the fungi, and then our core proteome of resistance or susceptibility, we're particularly interested in this category of proteins that show a significant change in abundance in the resistant strains versus the susceptible. And if we tease that apart and look a little bit further, here we have a list of 6 proteins that are commonly identified in both our resistant strains. So whether it's lab evolved or it's a clinical isolate, and they're significantly increased versus the susceptible. So in the volcano plot here on this side, these are stacked volcano plots. And so you have your lab evolved and your clinical isolate are present here, and then the wild type over on this side here. And those that are filled circles are common between the two comparisons. And one that immediately drew our attention because of its high difference between the resistant and susceptibility, as well as its function, was ClpX. And so ClpX is a unfoldase protein that is associated with proteolysis, as well as destruction of different proteins. It goes into a ClpX complex to degrade proteins. It also has roles in stabilizing proteins and misfolded proteins within the mitochondria. And so we want to look into that a little bit further. And so we did some different in vitro assays with a collaborator from Germany, for Dr. Stefan Sieber, and he was working on ClpX in bacterial pathogens and looking at inhibition of this target. And so we took our fungi, cryptococcal strains, and we treated with the inhibitor at varying concentrations. And as the increase in concentrations of the inhibitor for ClpX went up, the ability of the fungus survived went down. This didn't depend on whether it was resistant or susceptible. It was a general antifungal approach. If we then combined the inhibitor with fluconazole, then we started to see very quickly a reduction in fungal growth. And so we know that there's something happening between fluconazole, the drug that we're using to treat the infection, and inhibition of ClpX. So we then did a checkerboard assay to look for synergy, and we had synergistic effects between fluconazole and this ClpX inhibitor. And then we did in vitro assays where we took macrophage, infected them with cryptococcal strains, treated them with fluconazole, inhibitor, and the combination. And then we count the number of cells that come out from the fungal side. And we see that with that combination of treatment, we have a significant reduction in the ability of the fungus to survive. And so this was quite promising for us that ClpX does have a role in that resistance. So the next thing we wanted to do was move into an in vivo model. And to do that, we could use the inhibitor, the ClpX inhibitor. However, it was a topical treatment. And because Cryptococcus infects the lungs, we decided to go with disruption of the gene instead to mimic that disruption of ClpX. And so we made gene deletion strains, and in the absence of fluconazole, they grow the same. In the presence of fluconazole, we have our mutants that have a significant reduction in growth. And so we can see that difference between resistance and susceptibility again. And then if we do our mouse assays, in the first plot here, we have our lab-evolved strain where the survival of the mice ends around about 20 days, which is in line with previous experiments. When we have our ClpX mutant, it has a defect in virulence, and so we see a reduction in survival. But what was really exciting is that when we add fluconazole to this model, our lab-evolved strains still succumb to infection, but our ClpX mutants are actually able to survive to the end of our trial. And we had to keep pushing the trial end date further and further away because the mice were very healthy. And so it indicates that they're overcoming infection. So if we have a mouse that is infected with that ClpX mutant strain, or we disrupt the target, then the mouse is actually able to overcome infection and survive to the end of the trial. And so fluconazole is able to do its job and treat the infection. If we look at the fungal burden in the lungs and the brain, we see this mimicked, where we have a significant decrease in the fungal burden in both of those organs upon treatment. And so it was really exciting for us to see this and that we can make fluconazole work again within this model. So we wanted to then tease apart what is the mechanism of how this is working and what ClpX is doing within the fungal strains. And so we performed looking at different assays here, looking at percentage of ergosterol, which is the target of fluconazole. So you can see in the absence of fluconazole here, there's a difference between our lab evolved, but not between our mutants. When we add fluconazole to the system and you force that resistance, this is where we see our lab evolved has an increase in ergosterol production. So it is an increase in the target being produced of fluconazole. Whereas if we remove ClpX from the system, it mimics the wild type. And so this aligns with what we were seeing before within our in vivo and in vitro models as well. And if we also look at heme content, because heme is a driver of ergosterol production, so if we look upstream of what is happening here, we see that we have a really high increase in the lab evolved versus in the mutant and our wild type as well. And then if we look at genes too, the ERG11 and ERG10, and we look at the proteins that are produced, we see this similar pattern. And so then what we want to do is we want to say, if we remove ClpX from the system and we treat with fluconazole, we can kill the fungus. And so that's a win. What happens if we supplement back heme and iron so that these strains can produce their target? Are they able to grow? So can we actually reinstate that resistance? And so these assays here show you the susceptible strain unable to grow in the presence of the drug, the resistant strain that can grow all the time in the presence of the drug, and then our mutant strain where it is unable to grow in the presence of fluconazole, but we can actually, if we supplement heme and iron back in the media, it can grow again. So we have this ability to turn on and off antifungal resistance within this model. And so that built us to proposing a model of fluconazole resistance itself. Within our resistant strains, we have an increase in ergosterol production, which is the target of fluconazole. And we believe this to be driven by the presence of ClpX, which is able to stabilize this heme complex within the mitochondria. And then we get a high production of heme, and we get increased production of ERG11 and ergosterol, which is leading to that more target. So you're kind of saturating the membrane of the fungal cell to the drug itself. Whereas in the susceptible strain, in the absence of ClpX, our heme biosynthesis is disrupted. We do not have as much production of it, which then drives a lower rate of ergosterol, which makes the target, which reduces the target availability for fluconazole. And so this is why our cells are becoming susceptible to the drug after we remove ClpX from the system. And so working within this model system, what we want to do then is to develop new inhibitors or new drugs for ClpX in this particular example, but also for other targets as well. So we selected ClpX from the top 6, but it was just one of the ones that we chose. And so we have 5 other ones that we could work with. We got lucky by picking ClpX. I'm not going to lie. It was really nice that it worked because many times they don't work. So we want to be able to build this in an in silico approach. And so I have a PhD student, Carrie Woolnough, who is working on this novel inhibitor discovery approach, specifically towards ClpX. But really the goal is to develop a pipeline so that we can put in other targets as well and identify inhibitors. And because we already have an inhibitor for ClpX, we're using that as our positive control. And we're trying to design other inhibitors that would perhaps be more bioavailable within the lungs, and other ones that are different from ClpX, structurally different from through their compound too. And so the project is put into two aims. We have structure prediction and virtual screening as objective 2. And the different components, we have our target identification, which we've already done with ClpX. We then predict the structure, and Carrie has used AlphaFold for this structure prediction, as well as looking for homology in the protein data bank, and then looking at structure function analysis. This information is then put into our virtual screening method, and I'll go through those steps as well, where we're looking at building a library or using a library and narrowing down new compounds that we could test in the lab, docking those to ClpX to look at binding affinities, looking at molecular dynamics and the movement that occurs and the stability and off-target impacts, then moving into an in vitro validation, and finally optimizing some of our candidates. And so very briefly, just taking a look at some of Carrie's work here with the objective 1, the structure prediction, some of the initial challenges were the disordered regions. And so in an earlier version of AlphaFold multimer here, we have in red are the highly disordered regions that you can see these loops outside of the structure, as well as within the structure as well. And we have large gaps that occur between the domains of this homohexamer. And then also by a predicted aligned error plot here, we can also look at the accuracy of the relative positioning, and in yellow are the disordered areas where we really see this lack of consensus in what the model should be and look like. And so then Carrie has worked to improve the accuracy and also worked with homolog mapping. And so in more recent versions of AlphaFold that can handle more larger protein complexes, we have an improved confidence in packing of the domain. So you can see in our original here versus in this one here, we have more confidence and less disordered regions that we're able to then predict this model based, or to base the model on. And then to look at other proteins. So because the cryptococcal ClpX protein has not been crystallized and we don't have a structure for it, we can, through sequence homology, we can look at other comparable ones and then map onto it. And so that's what Carrie has done here, is a superposition of the predicted versus experimental ones of a homolog in a bacterial pathogen as well, to give us confidence in our model and whether what we're looking at is working or not. And then for the virtual screening for the library filtering and molecular docking, Carrie has worked with the Zinc 20 database, which has about 650 million molecules. And then the library filtering is based on likeness to our compound. So 334 is the inhibitor that we have from our collaborator for ClpX. And we've used that as a starting point to look for other potential compounds that have a similar mode of action or a similar structure as well, molecular weight. But that's just one of our filtering steps. And so depending on what we get from the data, we can always go back and remove this likeness and look at other properties as well. And so Carrie has then filtered it down to about 40,000 molecules and is using Gold and Autodoc Vina, using Compute Canada then to do molecular docking experiments to prioritize these candidates a bit further. And so then looking at, because she's using two different systems, the Gold and Autodoc Vina here, we have about 1,000 molecules that show improved binding affinity over our initial compound. And so we're optimistic that we're moving forward in our ability to identify novel inhibitors that could target ClpX, as well as in the pipeline itself. However, between the two systems, or between the two analyses here, we have poor correlation, which is something that has been well reported. And so there are different, I'll highlight here some work that was not from our group, but in 2019 that was published on how to correlate different analyses, so different docking outcomes. And this exponential consensus ranking allows you to look at ligands and decoys from different areas. So not just taking the top ranking from one, the top ranking from the other, or those that are ranked in both, but actually considering all the different parameters. And so this is what Carrie is doing then between the two platforms to really identify molecules that will be the best for us to continue within the lab and look at further. And so the next steps then is that Carrie is rerunning the molecular docking on the same library with the both programs to have a replicate of our analysis, as well as testing additional programs, and then performing molecular dynamics of these protein ligand conformations of the top ranked molecules so we can further narrow it down. And then we want to move these molecules and these compounds into the lab for in vitro testing to see if they really do have an impact on ClpX, if they're better than our initial inhibitor, and if we could move them into an in vivo model eventually. And as I mentioned as well, we start with those drugs that are the compounds similar to 334, but we can also then look at others as well. And so to move these, so we have our inhibitor compounds and our novel compounds that we want to look at, as well as our initial inhibitor too for ClpX. And a part of this is then to look at what are the impacts in the cell. So we know that if we disrupt ClpX, we're able to reverse the resistance to fluconazole, but how is that happening and why? And what are the off-target effects of this treatment? Because if we want to move it into a clinical application, we have to be able to show that it's not detrimental to the host, any more so than fluconazole itself. And so Michael Woods, a PhD student in my group, is now integrating different proteomics techniques to look at those on and off target effects. He's starting with total proteome analysis. And so he takes control treated cells and then the inhibitor treated cells and looks at the total proteome changes from those. He's also using PISA, which is a proteome integral solubility alteration assay, which is similar to thermal proteome profiling. And so you take your untreated cells and your treated cells, and over a temperature gradient, you look at the solubility of proteins. And the theory is that if they bind to your inhibitor and they're stabilized, they will survive longer at higher temperatures. And so your concentration will increase. And so for both of these sample sets, then he takes the samples, extracts proteins from it, and then we perform our proteomics pipeline and analysis to identify these proteins. And he has some preliminary data where he's looked at this PCA shows the difference between the treated and the untreated samples, as well as a column correlation as well that separates them out. And you can see in the volcano plot here, we have proteins that show a significant enrichment within the treated samples after that thermal gradient profiling, which allows us to identify on and off target effects as well. What is happening when you have that inhibitor? What is it binding to? And what proteins is it stabilizing within the system? And so in summary, from this study here, this component that we're contributing to is that fluconazole resistance drives proteome remodeling. And that proteome remodeling then gives us an insight into how we can actually overcome resistance. We can inhibit or disrupt specific protein targets to then revert to a susceptible phenotype for the strain. And we can use and apply different in silico drug discovery approaches to prioritize new compounds, as well as assess the current ones that we have for in vitro and in vivo testing with the foresight or the idea to move these into the clinic. So the next steps then are mechanistic characterization of the novel compounds, looking for those on and off target effects, evaluate different protein targets. So we have PRA1, for example, was on the list with ClpX. And so we've done similar experiments with that as well. And we see a reduction of resistance to fluconazole in that strain too. And so it's nice that it's not just a one-off, that we are able to do this at the protein level. And then also look at different classes of antifungals and pathogens. So we've applied this to Cryptococcus and fluconazole, but really there's many other fungal pathogens as well that have a much larger global impact that we can then still apply this workflow and strategy towards. And so with that, I also want to highlight Rochelle, our winner from the CNPN, ISMB Travel Award winner as well, and to let you know about the Canadian National Proteomics Network. And so if you're not a member, I encourage you to look it up and perhaps become a member. We have a different spotlight series where we really highlight work by mass spectrometry and computational mass spectrometry as well, and provide different opportunities. I also started Mums in Proteomics. And so if you are a mother in proteomics, if you are a supporter of Mums in Proteomics, or if you have a mother, you are welcome to join. And we have, it's an international community, and it's very much built on recognizing and supporting mothers in STEM. And lastly, I would also like to highlight Arnaud Dewa and I are hosting a Canadian Bioinformatics workshop in proteomics at Laval University in Quebec in the middle of September. And so if you're interested in learning more about proteomics and bioinformatics data analysis, there's still time to register for that as well. And I will have a talk in the Bioinformatics in Canada session tomorrow afternoon on the formation of the Canadian Canadian Proteomics Association Artificial Intelligence Consortium, which we have now built towards the Canadian Artificial Intelligence and Mass Spectrometry Symposium and Consortium. And so if you're interested in hearing about that, please join. Okay, that is everything. Thank you very much. I want to thank my outstanding group members, because without them, I wouldn't be up here being able to talk to you. And then our funders for the entire One Health program. And so we really do try to integrate what we do and our techniques and what we learn from the different areas within our research lab. Thank you very much.
Thank you very much. So we have a few questions.
Thank you, Jennifer, for the talk. We have one question from online. Given that ClpX can form a homohexamer based on your analysis, how was it verified that the inhibitor does not bind to the multimer interface? In case of bacteria, ClpX has been known to interact with ClPP before substrate recognition. Is this interaction with ClPP also observed in fungi?
Yes, so within Cryptococcus, the interaction between ClpX and ClpP is not well defined or observed. However, that question about the binding site is an excellent question, because the group in Germany, Stefan Sieber's group, has done a lot of analysis on where does the compound 334 bind? Is it at because they're working in Staphylococcus, and so it's the bacterial system. And so does it bind at the interface between ClpX and ClpP, or is it binding to the monomer? And they do not have a specific binding site yet. And so we have, Carrie has then done some testing to have it bind within the ATP pocket of the ClpX itself. We do not know if that's confidently binding in there, but that's at least a starting point for us. And then we can also look at other potential binding sites as well. So it's an excellent question. Thank you.
Very nice work. You mentioned that you're interested in finding targets that are, compounds that are a little bit more bioavailable to the lungs. My question would be twofold. Is it possible to, is there data, is it possible to quantify bioavailability to the lung in the first place? And could you incorporate that into your model to prioritize targets that would be bioavailable?
Yeah, absolutely. So we can do the tracking within like an in vivo system. We could track how much goes to the lungs depending on if we used labeled. For example, if we used a labeled compound or something, then we could track that. But computationally, so yes, so Carrie has done a very unbiased look at it. But the next round would then be adding in those pharmacokinetic and pharmacodynamic properties, such as bioavailability in different areas. I don't know if we could add in lungs specifically, but we could perhaps localize it in some way, or at least look at other ones and then use that as a parameter. So we haven't done it yet, but it is one of our steps that we can apply. Yeah.
Thank you.
Thanks, Natu.
Great talk. Quick question regarding your future plans when it comes to the mechanistic characterization of your inhibitor. Do you plan to also look at signaling pathways that might play a role? And if so, given the fact that you're already doing TPP-like experiments, have you thought about a decrypt-M-like approach where you treat with inhibitor and then follow, say, phosphorylation through the signaling network?
Yeah, it's a great question. I avoid phosphorylation at all costs. So no, we do some in my lab. We do post-translational modification analysis, primarily phosphorylation, and we've done it with crypto, and we've done it with antifungal, like with the fluconazole-resistant strains, but never with the inhibitor. So absolutely, that is something that we could do and track it over time, but not something we've explored yet. Yeah. Thanks.
Thank you, Jennifer, for this talk. I was wondering if there's a risk that these, inhibitors that you're discovering would also result in resistance later, and how do you deal with that, and whether this could be another prioritization criteria for the inhibitors that you're looking for?
Yes, absolutely. So depending on the target, resistance is always possible, and the evolution of resistance is always possible. The thing that gives us some promise or some hope is that if we can reduce the amount of fluconazole that we have to deliver, and we can, then maybe we can prolong the development of resistance. But against this particular inhibitor, it would depend on the target. So if the target is present within the human host and within the fungi, that's one of our criteria we use for selection, as well as how important that target is. So if there's redundancy, so for example, with ergosterol in the membrane, these cells can produce more. And so this evolution can occur relatively quickly to fluconazole. But if we're targeting ClpX and it's required, it may not be as easy for the cells to adapt it. And then it also depends on where the inhibitor binds within that protein as well. So all considerations, absolutely, but you're right, it is possible. Yeah.
Thank you.
Thank you, Julie.